So thank you very much, Claire, for the introduction, and good morning, everybody. We're really delighted to have the opportunity to speak to you today about the National Centre for the 3Rs. This is a really fantastic organisation that I've had the privilege to work for over the last 14 years, and I appreciate that for many of you, you may be familiar with aspects of the work that we do at the Centre, but I'm also equally aware that for many of you listening today, this may be your first interaction with us. So welcome to you all, and hopefully, irrespective of your base knowledge of us, you'll go away from today's presentation with a better appreciation of the role of the Centre and how we support better science and animal welfare through the application of the 3Rs. That's the replacement, refinement and reductions of animals in research. And it would be remiss of me without going further to just give a really big thank you to the MRC and the BBSRC for the continued support they have given us as our major core funders over the years. And I'm also delighted to share the virtual stage this morning with Dr. Nathalie, Percie de Sert, who you'll be hearing from a little bit later on. So before telling you a little bit more about the work that we do, I think it's important to put the animal research issue into a little context. So animals have been used in scientific work for centuries, and they've contributed to many of the medical advances we now take for granted in terms of vaccines, antibiotics, anesthetics, and treatments for diseases such as diabetes and high blood pressure. And animals continue to be used today, with over 3 million being used in Great Britain each year and many millions more being used worldwide. And they're used for basic research to generate new scientific knowledge, some of which may include better understanding of how the different tissues and organs of the body work when healthy and diseased, throughout the drug discovery and development process to support decisions on which drugs are safe and efficacious enough to be used in humans, to test commonly used chemical products to ensure they're safe for animals, humans and the environment. And also in the same way that animals are used to understand human health and disease, they're also used for studying diseases in animals and in the development of veterinary products. However, despite the benefits and the advances that come from animal research, and that will continue to do so, it remains one of the most contentious and complex areas of science that goes beyond the ethical issues of causing harm to animals, but also includes matters relating to research culture and integrity. It remains an area of high public concern, and every couple of years, MORI conduct a poll of public attitudes to animal research, which highlights that the British public is generally supportive of animal research, but this is falling, and that this support is conditional on the 3Rs principles being applied. Something that Nathalie will talk more about later are concerns regarding whether animal studies are appropriately designed and reproducible, and the impact that this has on the quality of the scientific output and the numbers of animals used. And finally, the scientific validity of animal models and how well they translate to humans is another area of concern, and one that regularly appears in journals and the scientific press. And there are now even whole Twitter accounts dedicated to calling out the hype and reporting of results generated in animals and how these may translate to humans. But the 3Rs gives us a mechanism to try and address these concerns by providing a framework for critiquing the utility of animal models, designing appropriate animal studies, and for responding to societal concerns about animal research. And these aren't a new concept. They were first proposed in 1959 by these gentlemen that you can see here, William Russell and Rex Birch, in their book, The Principles of Humane Experimental Technique. And they defined each of the Rs as replacement being any opportunities to avoid or replace the use of animals in areas where they otherwise would have been used. Reduction is minimizing the number of animals consistent with the scientific aims of the study. And refinement are opportunities for minimizing pain, suffering and distress or lasting harm that lab animals might experience across their lifetime. And since they were proposed, they've been embedded in national and international legislation and regulations on the use of animals in research. But despite this, there often lacks the drivers and the support for researchers to truly embed the 3Rs at the heart of their research. So in 2004, following a House of Lords review of the way that animals are used in the biosciences in the UK, the NC3Rs was created by government because they recognised the need for more scientific activity in the 3Rs, and we were established out of the MRC's own Centre for Best Practice in Animal Research. And this is a team that you can see here celebrating our 15th birthday in October last year. And I would like to take this opportunity just to recognise what a fantastic group of individuals that I get to work with that are both driven, high quality, and passionate. And it's because of them that the Centre's been as impactful as it has. And historically, there's been little activity from the scientific community in the 3Rs, and it's our job at the Centre to change that. So we've developed a strategy which includes funding research and innovation and a very large program of in-house activities which are led by our staff to drive changes in policy, practice and regulations around animal research. And we work internationally with industry across the pharmaceutical, chemical, consumer product and agribusiness sectors. academia, regulators and funders, and our remit is broad, including any area of animal use for research purposes, so right across biological, medical and veterinary disciplines. And our team is based mainly in London, but we also have a number of regional staff that are embedded within universities across the UK to support those organisations in applying the 3Rs on the ground. Our annual budget is around £10 million per year, and again, we're very grateful to the MRC and BBSRC as our major core funders. And the activities of the Centre are supported by an independent non-executive board. So as I just highlighted, our strategy is not only to fund research, innovation and early career scientists, but also to embed advances in the 3Rs in policy, practice and regulations through our in-house programs. And these programs cut right across the organisation's activities and are driven by our in-house team of scientific staff. And these are the examples that you're going to hear about today. So opportunities for the use of single species in drug development, applying the 3Rs in batch release and quality control testing of biologics, minimising aggression in group housed male mice, and finally our broad programme to look at experimental design. And the first of these examples is a project that we've developed as part of an ongoing collaboration with the Association of the British Pharmaceutical Industry, so that's the ABPI, to deliver 3Rs impacts in drug development, because companies have concerns about the use of animals and they also recognise that they may not be as predictive as they'd like, which presents a strong business case for them to find alternatives. And within the pharmaceutical industry, animals, including mice, rats, dogs and monkeys, are used across the drug development pipeline to test if potential new drugs are safe and efficacious by determining what effects these drugs have on different body systems and the potential of different drug doses to cause harm or toxicity. And these studies are required to be compliant with international regulatory guidelines. And this process typically takes between 10 and 15 years, and it's eye-wateringly expensive. So it can cost between $1.5 to $2 billion to take a drug through to market. And a lot of this high cost can be attributed by late-stage failures, which we see within clinical development, because by this point, a lot of the money has been spent on each of these candidate compounds that are not going to be recouped through sales. And one key factor in this failure is the lack of translation of findings from preclinical models, including animal models, to the clinic. And so there are really significant efforts being made across the industry to try and develop 3Rs and apply 3Rs testing and strategies early within this development pipeline to support more physiologically relevant human-based testing approaches and reduce costs and animal use. And to support the industry in these efforts, we've worked closely with the ABPI and the wider industry sector and regulators internationally to develop a really large program to embed the 3Rs in safety and efficacy testing across the drug development pipeline. And these papers that you can see here, which have been co-authored with industry and regulatory colleagues, highlight some of the output from these efforts. But one of the most important projects that we've worked on has been our efforts in reviewing the use of 2 species within drug development. And this project really challenges the current dogma and regulatory guidelines which recommend that toxicity studies in 2 species are needed to assess the safety of new drugs. And these requirements have been around for the last 40 years or so, but the industry has moved on considerably in this time, so these guidelines no longer reflect contemporary knowledge and best practice. So typically testing to determine if something is safe or not in humans is done in 2 species, either the rat and the dog or the rat and the monkey. And this can require a minimum of 64 non-rodents for each molecule being tested. However, for newer modalities, such as biologics like monoclonal antibodies, development can be scientifically justified in just 1 species. So the aim of this project was to really explore whether both species always add value, and if these opportunities to use only 1 species could also be applied to a wider range of drugs than is currently, without compromising human safety. And the project included the major pharmaceutical companies, contract research organisations and regulators from across the UK, Europe and North America. And in all, 37 organisations were involved in the project, with 18 companies providing unpublished data for 172 drugs from various drug modalities and stages of development. And unfortunately, I don't have time to go through the data in any detail, but what this has demonstrated is that there are opportunities to use 1 species for toxicity studies for a much wider range of drug modalities than is currently permitted within the guidelines. And furthermore, that where 1 species is permitted, so for biologics, for example, this isn't being applied. And one of the really interesting questions within the survey was a retrospective question asking, could the company have made the same decision to progress or stop the compound using data from just 1 species alone? And you can see from this pie chart that 2/3 of respondents said that yes, they could. But of course, this is always easier to do with the benefit of hindsight and with having the data from 2 species behind you. So the important step for us now is to determine how can we use this information to make decisions prospectively. And so we're continuing to work with the companies and the regulators to try and determine this. And for biologics, it's already written in the guidelines that you can use 1 species. So we're focusing on trying to understand why companies haven't used this more often previously. And we're also trying to use case studies where we can to share experience and best practice and to raise awareness of the opportunities and to build confidence in the opportunities that exist. And for small molecules, we need to develop a larger evidence base to really demonstrate in what conditions dropping to 1 species is appropriate without compromising human safety. And if you'd like to know more about this project and the outputs from it, they were published earlier this year in Regulatory Toxicology and Pharmacology. So hopefully what this example has demonstrated is that even if certain test requirements are written into regulatory guidance and recommendations, there are still opportunities for changes to support cheaper, more efficient and more humane drug and chemical development. And we've worked extensively with all sectors and established ourselves as a trusted partner. Through our honest broker role for sharing data, we've also changed regulations at ICH level for pharmaceuticals, OECD level for chemicals, and within the US Environmental Protection Agency for pesticides, providing opportunities in each of these to minimize animal use and improve animal welfare. And the last example I'm going to give you before handing over to Nathalie is a very new and really exciting project that we're working on with the World Health Organization, and which is being co-funded by the Bill and Melinda Gates Foundation. And the aim of this project is to review the animal testing requirements within WHO guidelines for the quality control and batch release testing of biologics, and to make recommendations for implementation of 3Rs approaches and strategies. Now, what you can see here and what all these images have in common is that these all depict biologic products. And these are a really diverse group of medicines that includes vaccines against tetanus, diphtheria and polio, for example, snake antivenoms, therapeutic blood products, and biotherapeutics such as growth factors and monoclonal antibodies like Herceptin, which has been used for treating breast cancer. And unlike conventional small molecule drugs, which are derived synthetically, biologics are live products which are grown and then purified from cell cultures. And so these have to be processed under very tightly controlled conditions and to be tested regularly following approval and before each batch of product can be released for use within the public. And the majority of these tests are carried out using animals, and there are WHO guidelines and recommendations which describe the required tests for each product, and which are used by the majority of biologics manufacturers and national control authorities worldwide. So as long as these tests are written within them, they'll continue to be used. And as with conventional drugs, animals, including rodents, rabbits, but also monkeys, are used at all stages of biologics development life cycle. And it's been estimated that around 10 million animals are used per year for this process. But the vast majority of this animal use, around almost 80% of animal use, is used purely for quality control and product batch release testing. So after all the development work has been carried out. And these can be really grim tests in terms of the potential for animal suffering and have been described by some as the most severe in drug development. But they're also expensive and labor intensive. And due to the high degree of variability, they can cause significant delays of sometimes for up to a year to the release of products for use by the communities who need them most. And many of these tests were developed in the 1950s and 70s, and they've not changed very much since then, despite significant advances in the science that we've witnessed in the interim. So there are real opportunities for applying new thinking and new science to these tests and to integrating 3Rs approaches. So what we're going to focus on within this project is to deliver a review of the animal testing requirements within WHO guidance documents for the quality control and batch release testing of biologics and vaccines that require the use of animals, and identify evidence-based recommendations for the integration of 3Rs approaches. We'll then present these recommendations for the WHO for them to implement within their guidance documents to influence how these tests are carried out globally. And the timing of this project is really important because there has been some progress in applying the 3Rs in this space already, but this tends to be piecemeal and at a local level, and change is really slow to occur. So for example, what this table shows is that within the European Pharmacopoeia, there are 3Rs approaches that are recognized for some animal tests, and some of these are starting to be adopted or explored by vaccines manufacturers. But these approaches are not similarly recognized by other regions. So if companies want to market their products in Asia, for example, those regulatory authorities may still require the animal tests to be carried out. And we're also seeing that other organizations and groups in this space are advancing 3Rs projects of their own, including the International Alliance for Biological Standardization and the Developing Countries Vaccines Manufacturing Network, two really important organizations within this space. So what we hope is that our new project with the WHO will build on these initial steps and harmonize activities globally so that we can really deliver a very significant impact. And we've got a pretty strong track record in this area, having funded some really transformative research and new model development in the way that vaccines and biologic products are developed and tested. And we've also supported international activities to facilitate the uptake of these approaches. And this includes in pertussis vaccines for whooping cough, clostridial vaccines, which are some of the most commonly used veterinary biologics, cell-based assays to replace the mouse bioassay in detecting botulinum toxins. But despite this and the progress that has been made in the area already, this will be no small undertaking. We're talking about more than 60 guidelines that are in scope of the review. But we have set the project up really well for success, and we're working with more than 20 different organizations, including manufacturers, regulators, and relevant networks and trade associations from across the globe to deliver the project within the next 3 years. And with that, I'd like to thank you very much for your attention, and I'm delighted to hand over to Nathalie, who will tell you about 2 further projects from within our portfolio of activities. Nathalie.
Thank you, Anthony. So I'm going to add 2 more examples to Anthony's presentation. And first, I'm going to talk about a project to improve the welfare of mice that are grouped housed in lab cages. And the problem that I will highlight is injuries that are caused by fighting, which are a real issue, as you can see from this poor mouse here, that has been beaten up by other animals in its cage. So improving animal welfare is an important part of NC3Rs, and we've developed many resources to help scientists and animal technicians improve the welfare of animals used in experiments. And that's from rodents and fish to non-human primates. And improving welfare is not only important to the animals, but also to the scientific validity of the experiment, because animals in pain or in distress do not make good models. Pain and distress may increase the variability of the experiment, which means that more animals need to be used to detect the effect of an experimental intervention. And pain and distress can also confound the results, as we might think that we're measuring a real effect, but we're actually measuring the physiological impact of stress. So at the NC3Rs, we try to focus on things that affect a large number of animals and look at what happens to the animals from birth to death. And these are just a few examples of projects that we've run and guidance we've developed over the years. So improving welfare does not necessarily require high-tech equipment, and often a simple thing makes a huge difference. So for example, the way that you pick up a mouse will influence their stress level and the way that they behave in the experiment and respond to treatments. So we've done a lot of work on mouse handling to support scientists and animal facilities to change their practice and pick up mice with tunnels or cupped hands instead of picking them up by the tail, because there's a lot of evidence showing that picking up mice by the tail causes stress and impacts on scientific outcomes. Another area where we can make a big impact is mouse aggression. So sometimes when male mice are housed together in the same cage, they fight. And that's because in the confined space of a small lab cage, they're not able to establish a territory and manage the social interactions effectively. So this is a scientific as well as an animal welfare problem, because aggressive injuries can be severe and even result in death. And the pain or stress induced by fighting may confound the scientific outcomes or increase the variability of the data. And mice may also need to be taken out of the experiment, so the data from these mice cannot be used. Housing the mice alone is not a solution, because isolation is also detrimental to the welfare of this social species. And also we cannot use female mice only in experiments because important sex differences may be missed. And this has a huge impact. 2.5 million mice were used in research last year in the UK alone, so a better understanding of aggression could benefit the welfare of many millions of mice worldwide. And there's been a lot of discussion about mass aggression. This is not a new problem at all, but there's little evidence to support a strategy to improve this situation. So to devise a strategy, we needed to access the expertise of animal technicians who are looking after the animals day to day. And the best way to do this was a crowdsourcing approach, which had never been done in this community before. And to identify the triggers for aggression, we collected data from animal technicians on incidents of injury related to aggression, and we also collected the husbandry conditions at the facility. So 143 animal technicians from 44 facilities in 9 different countries participated in the study, and over 137,000 mice in 45,000 cages were observed during the 4-week data collection period in 2017. So what did we find? First of all, we now have a hard number on the prevalence of aggression. The average number of injuries related to aggression was 15 in 1,000 mice. So this figure represents a background rate of aggression, which can be used by facilities as a benchmark of current practice. Then we were able to identify factors that either contribute to aggression or reduce it. So we identified important strain differences in the prevalence of spontaneous cage aggression. It's common knowledge that some strains are more aggressive than others, but the scale of the study allowed us to provide a clear comparison between multiple strains and showed that some strains previously considered aggressive actually showed low levels of aggression. And by comparing facilities with each other, we were able to identify those husbandry conditions that trigger increased aggression, so we could make recommendations on how to minimize it. And practices that were found to decrease aggression included reducing the frequency of full cage cleaning, transferring dry nesting material from dirty to clean cages to retain important scent cues. And scent is really important here because this is how mice establish who they're friends with. And establishing groups using littermates was also found to decrease aggression. So these findings were published in scientific reports last year, and our recommendations on how to minimize aggression are being implemented by mouse facilities worldwide, with follow-up questionnaires demonstrating real impact in terms of reduced aggression and improved animal welfare. So we can get metrics on what has changed as a result of our recommendations. And this was a very successful approach, so we want to use it again. And we're now planning an activity to look at how to improve the welfare of zebrafish, which is the second most commonly used species in research in the UK. So now we'll give you an example from our program of work on experimental design and reporting. And considering the sensitivity of animal research and the ethical and societal concerns with it, you would hope that animal experiments are carefully designed and transparently reported to ensure that the results are reliable and that they all add to the knowledge base and contribute to scientific progress. Unfortunately, the reality is quite different. Many scientists and organizations are concerned about the quality and the reproducibility of animal research. So these are just a few of the headlines from the last few years. And some are talking about a reproducibility crisis. And this movement was given a lot of momentum about 10 years ago when the pharmaceutical industry reported that they could not reproduce most of what was published in the scientific literature. And this is something that we are concerned about at the NC3Rs, because if studies do not yield reliable results, they can't add to the evidence base and it's a waste of animals. And there are many factors that contribute to the poor reproducibility of animal research. The main ones are that there's not enough rigor in the study design and the data analysis, which means that results are often unreliable. And when experiments are published in the scientific literature, there are not enough information to allow others to repeat the methods that have been used or to assess whether the results can be trusted. And this has a massive impact on scientific progress. It's very common to see treatments that worked in animal model fail at the clinical trial stage because they're actually not effective or because they're harmful. And this also has an impact on resources. It's estimated that half of all preclinical research is not reproducible. So in the US, the budget for preclinical research is $56 billion per year. So 28 billion a year are wasted in the US only. And if you apply this estimation to research done in the UK, this could mean a waste of 1.6 million animals every year. So in a rigorous study design, you would expect that a number of measures are used to reduce the risk of subjective bias. So just to give you an example, One measure that can reduce the risk of bias is blinding, so that investigators don't know what experimental intervention the animal have received and they can remain objective. So this study is over 50 years old, but it really demonstrates why blinding is important. This is an experiment that was carried out on two populations of rats, one described as bright rats and the other described as dull rats. And the rats were put through a memory test, a TMS, which had a white and a dark arm. And during the training phase, rats would learn to go to the dark side and they would get a food reward when they made a correct turn to the dark arm. Then the experiment was carried over several days, and the researchers counted the number of correct turns to the dark arm. So as you can see on the graph, the bright rats did significantly better than the dull rat, and they started off better and they improved further over the course of the experiment. The thing is, the experiment was actually not performed on the rats, it was performed on the researchers. And there was actually no difference between the two populations of rats. They had been labeled bright or dull randomly in the morning of the experiment. The only difference between the two populations of rats were in the minds and the expectations of the researchers who ran the experiments. Rats in the bright group were perceived as brighter, cleaner, tamer, they were more pleasant to work with, and rats in the dull group actually even beat one of the researchers. So the researchers working with the bright rat described their own behavior as more relaxed and casual. So these differences on the graph are actually due to differences in researchers' behaviors towards the rats rather than the rats themselves. And that's why blinding is extremely important, because if researchers are not blinded, they can unconsciously influence the results. And the example that I've just shown is very nice, but it has real life consequences. In experiments in animals looking at the effect of a new drug, for example, unconscious bias by the researchers will lead to an overestimation of the treatment efficacy. So the treatment may be taken to clinical trial when it should not have been. And NC3Rs is doing a lot to address this and improve the quality of animal experiments. This is an example of a tool we developed and launched 5 years ago called the Experimental Design Assistant. And this tool is an online system to help researchers design in vivo experiments. It's a very clever system which acts like your own personal statistical assistant. So researchers design experiments in the system, and it uses computer-based logical reasoning to provide bespoke advice and feedback on the experimental plans and the statistical analysis. So it provides dedicated support for randomization, blinding, and power calculation. It's helping researchers design experiments as robust and rigorous as possible, using the minimum number of animals that they need and addressing sources of bias with randomization and blinding to ensure that the results are reliable and reproducible. It also provides improved transparency for those involved in reviewing these experiments, like funders. And another resource that we have developed is the RAV guidelines, which set out the minimum information that should be reported in scientific publication describing animal research. So these guidelines have been established as an international standard over the last 10 years. And these resources are endorsed by many of the major stakeholders in scientific research. So for example, over 1,000 journals recommend the RAV guidelines in their instruction to authors. And many major funding organizations recommend that researchers use the Experimental Design Assistant when preparing a grant application that contains animal research. So that includes the MRC or the Wellcome Trust in the UK, but also funders in Europe and in the US, like the NIH. And there is also grassroots support from the researchers themselves. So the EDA, the Experimental Design Assistant, has close to 10,000 registered users. And over the past few years, we've had 40,000 requests for printed copies of the RAV guidelines from 50 different countries. So we really appreciate this opportunity to tell you about our staff-led projects today. And I think these examples really demonstrate the importance of the work that we do to embed the 3Rs into policy and regulation and to drive changes in practice. But funding new models and technologies that can reduce animal use or improve animal welfare is also extremely important. And over the last 15 years, at the NC3Rs, we've committed £100 million in grants, innovation, and early careers awards. And we recently published 3 documents that highlight the impact of the research that we found, both in terms of the 3Rs impact, but also crucially, its scientific impact. So if you want to know more, you can access these on the link showed at the bottom of the slide. And to finish, I'd like to thank all NC3R staff who led the project we've just described, and also the various individuals and organizations across the scientific community who helped support this work. And thank you to all of you as well for your time and your attention today, and to MRC for organizing this event.